Okay, great. Full disclosure, my formalized training is in pharmaceutical chemistry, but I have been involved in injectable generics since the beginning of my career, so I have had the pleasure of dealing with the microbiological side of this industry for over 27 years. And so, and the last 3 years of my industrial endeavors were in a regulatory affairs functionality where the majority of my job involved the interpretation and the application and the negotiation with the FDA regarding the most current guidances and regulations as applied to generic injectables. And so I'm very familiar with sterility assurance packages, ANDA submissions. Once we had approval for that section, how it used to be approved, you know, it was celebratory because the chemistry often went much more smoothly than the sterility assurance package was much more voluminous. So anyway, what I thought would be instructive is to take a look at the proposed 797, the draft 503B, and the current sterile fill CGMPs, and do kind of a diagram of how interrelated they are, and maybe point out some differences amongst the regulatory standards for these three, you know, closely related industries. So the sterility package really dictates the quality parameters, the critical parameters for products in sterility, endotoxin, and particulate matter. So let's dive right in. The ICH guidance on new products, etc., dictate that these are the quality parameters one wants to capture and validate and confirm in your sterility assurance package. So let's talk about really the limits of sterility testing. I believe Ross mentioned it. We're truly looking for a needle in a haystack on most occasions. You're looking for a statistical ability to determine a microbial contamination that often is related to a spurious event, etc. And I provided some quotes to illustrate this. It's again statistical. And most times, in my experience, you really don't encounter and capture sterility positives unless there's a gross contamination, because the majority of the units that you're testing will be, you know, sterile. They will be microbe-free. It's the few and far between from an event that occurred, and usually microbial contamination events are transient. It doesn't occur throughout the entire process. It's a one moment in time that something gets adulterated. And so you have no guarantees that one particular vial or that one particular syringe is the one that you're going to capture for sampling and testing. And so it's truly a hit or miss technique unless you have gross contamination. And I have also been involved where we have evaluated and performed quality CAPAs when there has been a gross contamination, and it often reveals itself in more than 30% of the product. So of course, then you know you have real issues with a particular event or seasonal event or room or something that went terribly wrong. So it's kind of the backdrop regarding sterility assurance. I prefer chemistry because it's homogeneous. You know, usually there's a cause and effect. There can be, you know, heat or acid or something that impacts your chemistry. But in microbiology, it's not nearly as evenly spread out or evenly eventful in your testing and product. Obviously, quality is something that is a culmination of your quality systems. All of your individuals within an organization need to understand everyone contributes to that ultimate quality product that goes to the patient, and it should be safe and efficacious. You can't test quality into the product. You can't fix it, etc., after the fact and test it into compliance. In testing, there's a couple of assumptions that we all subscribe to. Number one, that all units in the batch are the same or representative of that batch. Secondly, that the test results are assumed to represent the attributes of the remaining units you didn't test. I mean, that is batch release testing or compendial testing is performed on a sampling of the batch and not every unit because often it's destructive and therefore you wouldn't have any material to release to your patients. As I mentioned, microbial contamination is not uniformly distributed. It tends to cluster. It tends to be spurious and transient in nature. Fortunately, BET and particulate matter tend to be more evenly distributed and can signal problems with the batch or a lot of material. So the factors contributing to your sterility assurance package are all of these potential carriers of contamination. I have listed personnel in the top position because that is the entity that contaminates most of the time. It's what we call, it's the dirtiest element in the room, unfortunately, by nature of people and individuals. Air, water, equipment and consumables, components within the products, such as the active pharmaceutical ingredients, the excipients. If you're preparing a bulk formulation from active ingredients, excipients, fluids, those sorts of things, each one of those can contribute individually and collectively to the microbial integrity of your outcome, your bulk solution. And finally, your container closures. So the design element's relevant for sterility assurance, and you'll notice in one of the talks this morning, there was a lot of discussion around, or 483s around facilities. And the facilities design is essential for having and maintaining surfaces and environments that don't facilitate microbial growth. In fact, mitigate it or inhibit it and that sort of thing. The air and water cleanliness, environmental monitoring, personnel monitoring, these are all linchpins in maintaining a sterile environment as you go through your process. You collect your materials, you make your bulk solution, you fill it into your final container closures, etc. Now, the production and process controls, both in the individuals, their training, the material and process flow, and individual flow, you always want to be very cognizant of how not to introduce microbial contamination as the process of constructing or assembling the outcomes in your products. Your laboratory controls, your release and finished product and expiration dating, these are all sort of after the fact. After you've assembled your material, you do test it to confirm, hopefully, that you have no contamination and all of your systems have maintained a high quality product. And finally, systems maintenance. I think we touched on this. It's great to start up a new facility and have pristine environments, but maintenance of that environment is essential such that you don't incur rust, you don't have insects permeate your environment. You continue to have non-porous stainless steel that is very amenable to quality production. And then finally, what I call the policeman of our world, the quality assurance unit. Typically, people in industries will become complacent. It was kind of mentioned earlier where you get kind of comfortable in your environment. And so truly, it's the quality assurance enforcement of maintaining quality SOPs, monitoring the environment, ensuring people are following those SOPs, and that there's no signal via deviations or laboratory investigations or out-of-spec results that are suggesting some people are getting lax or not following what they're supposed to be following. And so that's an essential component of any organization in the pharma industries. Let's briefly talk about the facilities, and again, I'm trying not to read these. This is kind of collectively a resource to everybody. Layout, room separation, and process flow need to be thoughtfully incorporated in your construction parameters. They should be non-porous, cleanable surfaces. That's kind of like my retirement home. I want it to be easily cleanable, so I don't have to waste a lot of time doing that. Avoid sharp corners whenever possible. You want coved corners so that you don't provide a crevice or some surface that an organism might find, attractive. This includes making sure that you tightly marry surfaces for the installed components of the room. HVAC vents, pass-through compartments, windows. You really want all of that to be seamless in the eyes of an organism, so they can't see anywhere to find a niche and hide and that sort of thing. Materials should be resistant to deterioration from cleaning agents. You need to clean these surfaces. You need to wipe them down. You need to apply some really relatively caustic materials. And so, you really need to identify the polymers and the construction materials that will withhold numerous and continuous cleaning and disinfectant agents. And then clearly, you need a proper HVAC system to handle the air, keep the temperatures cool and the humidity low. Clean air, you mitigate your introduction of viable and non-viable particles. You cool temperatures, you inhibit the growth of organisms if they do happen to be there. And low humidity also. The drier the environment, the less attractive it is to microbial growth. And then finally, the concept is you want your cleanest area to have the highest quality of air, and then as you move away from that, you want the cascading air quality to be achieved on an inward to outward basis, and achieved typically using HEPA filters and unidirectional flow. So again, quickly, typically the critical zone is an ISO 5, contains not more than 3,520 particles, and then typically your surrounding or supporting areas are either ISO 7 or ISO 8, and all of these, there's a couple factors of increases in terms of 352,000 and then 3,520,000 particles, and those are astoundingly large numbers, but it is within the appropriate guidances that these rooms contain at or less than those particles. And it's usually achieved by properly installed HVAC and HEPA filters, such that your return vents are in appropriately positioned areas and your HEPAs are functioning optimally. So your first order of concern when you qualify your cleanest area, your ISO 5, is the airflow and quality of air in that particular region. And so in each of the subsequent slides, I've kind of identified for you the proposed regulatory standard, what they require, and any particular verbiage that I thought would be relevant and interesting, and maybe create some contrast and comparisons between the various regulatory standards. So for the proposed USP 797, they're saying you need to qualify that room. You run an in situ smoke study initially and every 6 months thereafter, and repeat that when you make changes that are substantial to that area. And it's performed under dynamic conditions. 503B GMPs, it's initially repeated and redone when changes are made to the critical zone. And it's under simulated operational conditions. As with sterile fill facilities, similar requirements. It demonstrates unidirectional flow because you do not want turbulence, eddy flow, stagnant air. All of those perturbations from good quality airflow could facilitate microbe growth. And I've been involved in some where the agency really likes to see videotapes of your smoke studies. First of all, to verify you did it, and second of all, to really see that the room has been characterized properly, and it provides a baseline when you go to make changes to the room, introduce different equipment, that you're maintaining and still have the integrity of that smoke study, providing you the validation that they're looking for. So the HEPA filter performance is another parameter during qualification of this ISO 5 critical zone. It's done initially and every 6 months for each of the 3 regulatory categories under dynamic conditions. And that testing includes the integrity of that filter, the particle counts created by the action of that filter working well, and then air velocity coming out at 6 inches from the HEPA filter. And then as a company, you define the distance close to that surface in the direct compounding area or in your laminar flow hood or in your sterile core. And it's driven by your internal SOP. And finally, you have to meet the particulate count on a regular basis across all 3 industries, and initially and every 6 months under dynamic conditions for USP. 503B initially and daily during operations, and then sterile fills initially and continuously during operations. So it's interesting to me that that room needs to be monitored for USP only once every 6 months under dynamic conditions. In a microbial world, that seems a little subjective, but that's what the USP proposed 797 is recommending. In these supporting zones, often the way your facility is laid out is you have the critical zone, which is an ISO 5, you have buffer, gowning area, or also an ancillary area where you may be doing some other manipulations. And those areas, smoke studies are silent in terms, there's nothing mentioned in USP and 503B CGMPs. It's recommended for sterile fills under dynamic conditions. But again, these are not required per se. For the HEPA filtered air changes in that room, it's interesting to me that USP 797 is very specific about that. Not less than 30 hourly HEPA air filtered changes during compounding activities in an ISO 7 region, and not less than 20 in an ISO 8. Whereas the 503B facilities is silent to that particular room characterization, and sterile fill facilities have recommendations, and they need to be defined by your SOP under dynamic conditions. So they leave it up to the manufacturer to characterize that parameter in the room. And finally, you still have to meet the ISO 7 or 8 particle count requirements for those regions to maintain it as a 7 or 8 ISO condition, initially and every 6 months for USP, initially and every 6 months for 503Bs, and initially and continuously during production for sterile fill. Relatively the same, except sterile fills require it during production. So now you've qualified your interior room, you've qualified your supporting areas, and now how do you maintain that room? Well, your environmental monitoring describes how you can ensure that the character and how you've set up that room and qualified it maintains in a state of control so that you can understand your product is safe from microbial contamination. So one of the controls is the environmental monitoring, and you monitor your non-viable and your viable particulate, initially and defined by SOP for USP, daily production for 503B, and sterile production fill continuously during production, and very similarly for the viable air particles. What about your surfaces and your differential pressures? All 3 regulatory standards require that you monitor your surfaces, and all require that you monitor differential pressures between your rooms. Because again, the goal is cleanest air in the center, and as it flows out, that air can get a little dirtier and a little dirtier. But you do want your ISO 5 or your sterile core to have the highest quality air generated by your HEPAs. Your room differentials are characterized every 6 months in USP, continuously during production for both of the CGMP standards. Your temperature and humidity in your environment is monitored in all 3 standards, daily during production for USP and 503B, and continuously during production for your sterile core. So again, in your USP and your 503B, you take 1 temperature throughout the day and you've met the minimum requirement for the guidances, your temperature and humidity. Most facilities, you would clearly want to have an ongoing continuous monitoring system just so you ensure that you're meeting those requirements, because microbes will grow if you don't maintain low temperature and low humidity. So to follow on from environmental monitoring, so now you have a solid core, you've got a solid understanding of your supporting area, you've been monitoring the environment to ensure the conditions for microbial growth are mitigated. Now you have individuals entering into that area and that region. How do we ensure they're not introducing contamination into your products? So we have a personnel monitoring program. They monitor, they should be gowned up. There's a subsequent slide about gowning qualification, but you're tested in terms of your aseptic techniques via gloved fingertips and thumb, and all 3 of the standards require that testing. Initially and every 6 months for the USP operators. 503B, it's initially and daily per shift during or immediately after production, and the sterile fill daily, initially and daily during production, and in association with each lot. So again, USP is rather infrequent. So you qualify an operator, and 6 months later you qualify them again. So during that 6 months, you really have no idea of their aseptic technique and their capabilities. And I'm not clear as an organization, I would be comfortable with only checking someone every 6 months. Critical sites on the gowns. As you gown up, the USP is silent about that. 503B says, yes, you define it. What are the appropriate locations on that gown that you're wearing during aseptic manipulations? And again, for sterile fill, they also indicate initially and at appropriate samplings per your SOP that you create for that activity. So now you have an idea of the room, the supporting rooms, your environment is in control, your personnel are well trained, that you put them on a qualification program, they understand they have to maintain their sanitary and sterilization practices. So now what about the equipment that might come in the room that you need to utilize to either fabricate or create your bulk solutions and your finished goods? Proposed USP says the surfaces should not be reactive or absorptive. You don't want any porous surfaces that microbes could attach to and grow from. The 503B, you have contact surfaces again. You don't want them to be reactive, additive, or absorptive, and it requires sanitization and sterilization. So you need to clean and sterilize for 503B. And for sterile fill, the equipment, usually it's a production skid or something, so the components are initially autoclaved and then assembled. So you need to demonstrate that you have validated procedures for sterilization, transportation, and assembly of that production skid prior to production of your sterile product. For consumables, I consider those materials that are one and done, sterile sets, sterile filters. There's even some larger liners for tanks that are used once and then discarded so that you don't have to worry about cleaning or contamination on secondary use of those. The USP indicates that surfaces and contact components are not reactive or absorptive and must be sterile prior to use. So again, it may be sterile as you purchase the package, but then handling of that will be essential for maintaining its sterility. In the 503B facility, sterility and endotoxin can be acceptable per vendor C of A, but it should be evaluated after it's been used to ensure that nothing has cracked, broken, or presented itself with an avenue for microbes to be introduced into your product. And the sterile production facility seldom are one and done materials used. So your containers and closures. So you have your great facility, you have great environmental monitoring, your personnel have been trained and are appropriately handling aseptic processes. You have a bulk batch that is now sterile, and now you deliver it or you fill it into your containers. So they need to be evaluated in all three regulatory standards. USP allows for the visual inspection of container closure integrity in the absence of testing. 503B and sterile fills, you need to demonstrate through USP 1207, which is your container closure integrity testing. Sterile production, you actually render your containers and closures sterile, typically through deep hydrogenation tunnels and sterilization procedures, all that need to be validated. The 503B world, typically you're filling into sterile devices, syringes, bags, cassettes, components, and so you just have to demonstrate after the fact that they are going to retain their integrity through CCIT testing. Further to containers and closures, the sterility standards proposed by USP, 503B, and sterile fill. The top one is the container closure must be accompanied by a C of A. Sterile and deep hydrogenation parameters must be part of that specification, and they must have passed those parameters. If you perform it on site, the process must be established and documented. For 503Bs, the enforcement discretion allows that if supplier certifies and labels the material ready to use, that if packaging integrity is verified upon receipt, that it hasn't been compromised or peeled apart, and there's a valid C of A, and it meets all the acceptance criteria, including sterility and deep hydrogenation, then it can be used without further testing. In the sterile fill world, you have to validate that everything you have rendered sterile and non-pyrogenic is in fact that, through testing and through protocols. So components, and in this case I'm being specific to APIs and excipients, there is an evaluation in each of the particular USP 503B and sterile fill facilities. The criteria for USP is that it complies with the USP monograph, the CFA with specifications, and passing test results is part of your documentation, and that that material, that API, was obtained from a FDA-registered facility. That's kind of where the USP is requiring the FDA-registered facility, which there was made mention of manufacturer versus supplier, whereby the manufacturer needs to be FDA registered despite the supplier providing that to you. They're just kind of the broker. In 503B, there is some discretion where you can establish a quality agreement with the supplier, and therefore there's at least 6 or 8 criteria. If you meet, you'll need to take a look at the guidance itself, but it speaks to your ensuring the integrity and the quality of that supplier and the materials they are providing you meet your requirements for quality. And once you've qualified them and you have a standing quality agreement, then you can receive materials with periodic testing and not full testing. In the sterile fill world, typically we source an API from under what we call a drug master file. And organizations that provide us that drug are audited by the FDA, their DMF, at time of our approval for an ANDA, and it contains just a plethora of requirements such that that API is from a registered FDA facility, and it has all of the appropriate documentation, validation, etc. contained therein. All the methods are validated and everything. Okay. Continuing on to components, if you're going to start from a source finished dosage form, then you do need to evaluate that as well in the USP and the CGMP world. Obviously, the sterile fills, they're creating this FDF, so there's no requirements because that is the product of their outcome. In USP, you need to comply with the USP monograph, C of A with specifications, passing results, and obtained from an FDA-registered facility. There is some allowable FDA enforcement discretion for 503Bs. The source drugs can be found acceptable without testing if they're purchased directly from the registered manufacturer, the label examined and it's verified that the product quality, and no portion of the lot is subject to a recall, and then finally that during shipment there's been no compromise of that container, so it does appear intact. So, is there a requirement for the endotoxin or bioburden assessment for components? For USP 797, it's not applicable. 503B and sterile fill, yes. And you typically purchase sterile deep hydrogenated components for under USP 797. That's allowable. Incoming materials and non-sterile components must include microbial and endotoxin testing as declared by the accompanying C of A. In the sterile fill area, you actually characterize that amount of bioburden or endotoxin of each component. So again, a slightly more aggressive approach by the sterile fills. So finally, I'm going to just talk about the production and process controls, because now you have your room, you have your personnel, you have your components, you have your individuals that are trained, and you've generated, and you're in the process of creating that bulk batch. So in essence, there's a number of sterilization and process controls that contribute to that maintenance of sterility. So I'm going to quickly go through these. Here we talk about the gowning annually, qualification initially. And again, you can see here there's a number of surfaces that need to be covered of the individual handling the production procedures. And there can be no exposed skin. 797 historically allowed you to have some exposed skin. That's no longer the practice. And specifically inside the ISO 5, nor donning or doffing gloves. The reuse of garb, in fact, USP has a caveat where you can use it, but it's a very specific situation as listed here. So we briefly talked about what is the role of media fills in qualifying analysts or operators. And it's very interesting because the proposed USP, you only need to qualify and complete one successful media fill. Both in 503Bs and in sterile productions, you have to do 3 successful media fills. And that means if you do 2 successfully and the 3rd one fails, you're starting all over again at number 1. And I've been involved in locations where it took several tries to get 3 successive media fills completed. So it's a bit more stringent to have that 3 successive criteria. Cleaning and disinfectant, we talked quite a bit about that. The roasted, there's a difference between disinfecting and sporicidal and cleaning. And so USP requires that only the sterile IPA is sterile. The other components can be non-sterile. And then it indicates, actually, I found it very interesting to, after you've cleaned, to wipe down with sterile IPA and 10 minutes later wipe that down. So you actually eliminate all your residues from some of your cleaning and sporicidal agents. Efficacy studies for cleaning and disinfecting. It's implied in the USP by the language that's in that 797 proposed guidance. It is required for both 503B and sterile facilities that you not only clean, but you demonstrate that the cleaning is efficacious and works. Your laboratory controls, is the product tested? USP 797, yes and no. None is required for Category 1 testing. And if you are going to test for Category 2, their only requirement is that you test for endotoxin and sterility, and they must comply with the USP 71 and 85. For 503B, you have to follow your appropriate procedures and test according to your requirements. Sterile fill, there's an entire page of regulations that you have to comply with to meet and approve and release product. Release or finished product testing. USP 797, again, yes and no. None required, but if you want to opt for sterility and endotoxin, you can extend your BUD from 4 days to 30 days at controlled room temperature. 503B facility, they've listed a number of parameters they'd like to see for testing that has been completed on those products. And then for sterile fill, you have an entire certificate of analysis, a set of specifications, and you need to test according to that. So I'm sure everybody kind of understands what this is, but I thought I'd put it in there. Beyond use date and expiration date are not interchangeable. Beyond use date means that the hour or the time in which is based on the initiation of production of that CSP, you cannot use it beyond. Expiration dating is the time during which the product you believe that you've fully tested maintains acceptability against those specifications and is under the specified storage conditions. And again, this is just a highlight of how do you assign the dating. All of these products require dating, and there's a complex decision-making section in 503B. I don't know if anybody's reviewed it lately. There's multiple tables, there's multiple conditions, there's, you know, if you did this, then this, if you did that, then this. So it's rather complex, and I'm not sure anybody has done a tree diagram or decision diagram for that. It might be very, very instructive. Yeah. And finally, I kind of mentioned this quality assurance surveillance or enforcement. There's a new term called a designated individual in 797, and they are actually defined as responsible and accountable for the performance and operation of the facility, and they must ensure that the facility has formal written QA and QC programs. Now, 503B and sterile production go a bit further, and they say there has to be a quality control unit that has responsibility for this, and that it should be an independent group within the corporate organizational structure to preserve the integrity of the quality group's function. And that has to be maintained, because if it's under the operations umbrella, then again, you're conflicted when things go awry and you're not going to make those production dates, or when you're not going to be able to ship product when something has gone wrong and quality has to put a hold on it. So these are the source documents, and that concludes my analysis. So thank you. Bye.